Hello, we are here with Chapter 17, the Special Senses. So we will be going over olfactory, which is smell, gustation, taste, vision, equilibrium, and hearing. First, we will talk about olfaction, our sense of smell. So this is where we are collecting chemical data to process as a sense of smell or an odorant. So the human nose, we contain about 10 to 100 million receptors, and these are in the olfactory epithelium that is located in the superior part of the nasal cavity that is on the ethmoid bone. The olfactory epithelium is covering the cribriform plate, the inferior surface of the cribriform plate of the ethmoid bone, and extends along the superior nasal concha. So that is going to be seen here, right along this area. Okay. 3 types of cells are going to be found in that olfactory epithelium. Olfactory receptor cells, detecting the odorant, supporting cells, and basal cells, or basal stem cells. So those olfactory receptors are going to be collecting those odorants coming into the nasal cavity. Once it triggers a response from the neuron, it sends a signal through that cribriform plate into the olfactory bulb, which lies on the superior surface of that cribriform plate. So here we are showing cranial nerve 1 with the olfactory receptors. It is not one single nerve. We actually have multiple areas of bundles of neurons moving through that cribriform plate, and they will synapse in a collective area in the olfactory bulb. From there, the signal will travel down the olfactory tract, where it will be received in the temporal lobe. Okay, so the supporting cells that we find, these are going to be columnar epithelium, located in the mucous membrane lining. Okay, mostly for physical support, nourishment, and we are making sure we don't have too much crossover with the olfactory receptors, so it is working as electrical insulation as well. And then the stem cells, these are undergoing mitosis to simply replace any wore out olfactory receptor cells. This is a good way to not lose our sense of smell. Obviously, as we get older, these basal stem cells do not tend to work quite as well. Olfactory glands, or Bowman's glands, these are producing mucus. They're going to help dissolve those odors that we are breathing in, that are going to help with transduction, which is conversion into electrical impulses. Okay, so the pathway that we are going to take whenever we detect a scent is going to go through the cribriform plate, the olfactory bulb, olfactory tract, and then the primary olfactory area of the cerebral cortex to the temporal lobe. That's seen here. So we breathe in, we detect a chemical odorant, we transfer that to the olfactory bulb. This moves to the temporal lobe. Now, we learned that most sensory information, except for smell, moves through the thalamus. Obviously, this is a clear vision of where it moves to instead of the thalamus. Okay, so with the olfactory transduction, we're detecting odorant and binding, and then this is moving to olfactory receptor protein. We are moving along by cyclic AMP, causing the depolarization So that odorant causes the neuron to depolarize. And then we have the action potential to the temporal lobe. And then we will also have frontal lobe communication for odor identification. So the temporal lobe senses that a smell has been detected, and the frontal lobe will identify that smell. And this is a picture of what's happening. And that is smell. Alright, our second special sense is gustation, or our sense of taste. This is going to take place mostly on the tongue, but we will see different areas of the mouth where we can detect taste. Okay, so these are simple chemical senses. We will have a chemical that causes depolarization of a neuron, telling our brain we have sensed a taste. We have 5 primary tastes: sour, sweet, bitter, salt, and umami, or meaty and savory. Okay, and we can have any number of combination of these. Okay, so taste buds. These are containing receptors for sensation of taste. So just like we saw olfactory epithelium for smell, we have gustatory receptor cells for our sense of taste. And these cells are being housed in the taste buds. Okay, so we have approximately around 10,000 taste buds on the tongue, on the soft palate, pharynx, and epiglottis. Okay, so we have 3 kinds of cells again. We have supporting cells, the receptor cells, and some more basal stem cells. So here we go. We're going to see taste buds. These nice small little areas found within papilla. Okay, so this taste bud shows the gustatory receptor cells. We have some supporting cells, and then basal stem cells. Okay, then we have the sensory neurons that are going to carry that signal to the brain. So real quick, back at the taste buds, you can see they are housed within another structure on the tongue. And this is papilla. We have 4 different types that we will talk about. Okay, so these taste buds are not just on the surface of the tongue, they're actually housed within different elevations called papilla. Okay, 3 types will contain taste buds, 1 will not. So we have the valate papilla. These are the larger papilla with quite a few taste buds. And then we have fungiform, kind of scattered all over the tongue, about 5 taste buds each. And then foliate papilla. Most of these are going to disappear after childhood. So these 3 contain taste buds. The one that does not is filiform or filiform. I like to say filiform because it reminds me that these papilla detect tactile sensation, so they feel what is on the tongue. No taste buds, but tactile receptors. Okay, so here we can see the disbursement of those papilla. Okay, so 3 cranial nerves are actually involved in our sense of taste. We mostly mention facial and glossopharyngeal. Facial is going to be the anterior 2/3 of the tongue. Glossopharyngeal will be the posterior 1/3 of the tongue. And then the vagus is going to pick up all those extra areas, the epiglottis and the pharynx, or in the throat. So these 3 cranial nerves are involved with taste. Okay, as you can see, taste is moving through the medulla, into the thalamus, then into the primary gustatory area found in the parietal lobe.